Friends, this video carries a short description about the cytoskeleton. So it is parted into two, that is microtubules and the microfilament structure and their dynamics, and the role of kinesin and tannin in the intracellular transport, which we will see in the next video. So cytoskeleton is an intracellular network of the protein filament present in the cytoplasm. It may be of the three types, that is microtubules, microfilament, and the intermediate filaments. So microtubules is made up of alpha beta tubulin heterodimers, and these protofilament align in a parallel with the same polarity, as in the figure clears the all the description about the microtubules, that it is made up of 13 protofilaments, and it may be the triplet or the doublet microtubules. So MTOCs are the microtubular organizing center, are the nucleating center that serve as a template for the polymerization of the tubulin. So these are the highly dynamic structure, constantly forming and disappearing depending on the cell activities. So the process of the polymerization assembly and the depolymerization disassembly of the microtubules appears to be a form of self-assembly. So colchicine and the vincristine vinblasting are the some common microtubules inhibiting drugs. So function of the microtubules are as follows, like form the structure framework of the flagella and the cilia, position of the cell organelle within the cells, direct the pattern of the cell wall synthesis in the plants. So, so this figure describes the structure of the centriole or the centrosome. So second one is the microfilament, which is made up of the actin filament made up of G-actin. So each G-actin contains a magnesium ion complexed with either ATP or the GTP. So figure clarifies the or all the description about the microfilaments. So there is a troponin and the tropomyosin over there, and the G-actin, that is globular actin, and the 2F-actin filamentous actin. So the polymerization of the G-actin occurs in three sequential phases, that is nucleation phase, elongation phase, and a steady state phase. So function of the microfilaments form the contractile ring for the cytokinins, shape of animal cells. It supports cell structure, microvilli. So example are the muscle contraction, microvilli of the brush border of the intestinal epithelium, and void movement, and the cytoplasmic streaming are the prominent. So last one is the intermediate filament, which is a rope-like and doesn't participate in a cell mortality and shows species specific variation. So monomer of the intermediate filament protein. So it has an N and C terminal, non-alpha helical head domain, alpha helical central domain, and the non-alpha helical tail domain. So intermediate filament is categorized into major six types of the uh, intermediate filament, that is acetic keratins, neutral basic keratins, vimentin, neurofilament protein, nuclear lamina, and the nestin. So function of the intermediate filament is the support to the cells and its nucleus. It forms a transcellular network to resist external forces when maintain filaments around and support the large fat droplets into fat cells. So this is about the cytoskeletons and their types. Thank you.